session. That is a continuous
Sure.
process.
Yeah.
So,
Dr. Tiku is there?
Dr. Aparna?
yeah, I called him. I just called him. He is joining in a minute. So, yeah,
Okay.
he is. Nataraja, so you were saying something?
Can you hear me? No problem?
Yes, yes, I can hear
Yes,
you very well.
Nataraj, we can hear you.
Thank you.
Perfectly
Thank you,
fine.
sir.
So, yeah, very, very good afternoon and good morning to Dr. Kasi. And welcome you all to this 47th webinar of this plant breeding webinar series, which is organized on a monthly basis by Foundation for Advanced Training in Plant Breeding. And today we have Dr. Kasi Azhakanandam, who is going to talk about challenges and solution for food security, nutrition, and environmental sustainability in a global south. Before the webinar, we are delighted to launch the new book authored by Dr. Nataraja Karaba. The purpose of this webinar is to highlight the importance of plant breeding while creating awareness on technology integration, policy support, regulatory framework, farmers' perspective, and inclusive crop improvement strategies. ATPBR is a non-profit organization dedicated to strengthening agriculture capacity, especially in plant breeding. With its global reach, ATPBR fosters skills, strategies, and collaboration that drives crop improvement. So far, hello, somebody saying
Please go ahead.
Okay. So far, we have organized 6 national conference, 2 global conference, 35 professional trainings, and 46 webinar, and more than 25,000 students and professionals attendees to our activities. We have published 10 books, including our flagship title, Market-Driven Plant Breeding for Practicing Breeder. Our training program have encouraged professionals from more than 100 organizations across 15 countries, and ATPBR hold 12A ATG and CSR certification, making us eligible for industry CSR project. Now, I take a privilege to introduce the moderator, Dr. N.K. Krishna Kumar, former DDG Horticulture Science, ICAR, and former regional representative, Biodiversity International. With over 40 years of service, his contribution to agriculture, horticulture, biodiversity, and research management are invaluable. He has authored more than 100 research papers and several books of national and international repute. He is a very active, very versatile, and very dynamic personality. Thank you, sir, for accepting to be today as a moderator. Now I request you to please take the floor. Sir, you are on mute. Please unmute.
Thank you, Aparna. Now I'm audible, I guess.
Yes, sir, please.
It's a privilege to always hear good words from Aparnaji, and also to see such eminent persons like Dr. Kasi, and also my good friend Nataraja Karaba, and so many other dignitaries who are here. I would. It's very appropriate that today's talk will focus. I'll first come to the talk and then introduce the speaker. Then we'll take a few minutes to release the book of Dr. Nataraja Karaba. Is that okay, Dr. Aparna? Okay.
Please, sir. Yes.
Yeah. So today's talk will be basically on challenges and solutions for food, nutrition, and environmental sustainability, with a special focus on the global south. So this topic is, I was just going through the topic, and it's a topic a little unique. For example, it's one of those things which is focusing on the global south, where 90% of the poverty, you can say that sustainability becomes a question, nutrition is a question. So the question of the food security and sustainability in the global south is an aspect which need to be considered, especially in an era where the global north is not so much worried about the global south, as we can see from the administrations of Donald Trump. With the due excuses to Dr. Kasi, who would probably agree with me. And we are going through a very transitional world where the challenges and solutions are not just limited to agriculture, science, and innovations, but very, very more also to the policy issues that govern the wide gap between the north and the south. Many of us understand the word nutrition in a very, very different way. Nutrition and nutrients are very different. Nutrition is a process by which an organism receives and utilizes the energy necessary for growth, development, and maintenance of its component processes. And rightfully, when Nataraja Karaba is there, nutrition focuses most on the physiology of the uptake and digestion and utilization of the nutrients, not per se the presence or absence of a nutrient in a particular system. So we always talk more in terms of nutrients, but the nutrition is a different cup of tea, or whether she's a lactating mother or an aging population, which is becoming more and more relevant. Environmental sustainability is another, it's not just a buzzword, it's a word which is something each and everyone has to be aware of in this world where the excessive use of agrochemicals or the dearth of pollinators, the microplastics in the environment. I mean, you can go endlessly, and it is the water table in the less developed world or the global south, and the diseases that are associated with it. All these things contribute to environmental sustainability. So, when we are talking about sustainability, it's neither agriculture nor nutrition nor environment, it is a melting pot of all this. So, very excellent topic, and I look forward to that. Coming to Dr. Kasi Azhakanandam, one of the first persons who I was happy to see that he has served in Mexico, he has served in Syngenta, he has got a tremendous number of patents. At least I could see 10 patents. A man who has worked in depth into the molecular aspects and the applications, especially on weedicides. He is a strategic consultant to bridge the R&D gap between biotechnology and sustainable agriculture. He was very heavily involved in the crop transformation lab and weed management at the molecular level. And I I think each one of the subjects that if you just see, each one is a profound contribution to our understanding of the sustainable development. We are all thinking about the weedicide resistant corn that we are talking, and a lot of discussions are taking place, whereas Dr. Karaba will be thinking more about microbiome and microbial populations. Somebody will be talking about economics. All these things are very, very important, and I look forward to the talk. And I have with me my very close friend Nataraja Karaba, whom comes from the laboratory of Dr. Krishna Shastri and Dr. Kumar, who probably the most leading physiologist, Dr. Uday Kumar, the most leading physiologists that this country has ever produced, who have transformed the lives of hundreds of scientists and educated hundreds, who are even today, some of whom are retired and still grateful to these people. He comes in that particular tradition. We both served in different assignments, especially in the sericulture biotechnology, and he is a person whom I admire. And today, his book on hologenomics insights, exploring the impact on crop physiology. It's a very beautiful cutting edge aspect of science that we are talking about, especially when we are talking about genetics and crop improvement. What is the apparent genetics of a particular plant? What is the phenotypic expression? We always talk about G into E, but when we are talking about E, the aspect that is pertaining to the microbial interactions, the way microbes facilitate both in the total expression of the phenotype, the disease resistance, the adaptability, and their radiation with regard to their adjustment in the environment. I think there are elasticity, everything. In fact, I would say that the whole world is a dance of the microbes, and we have not understood the dance of the microbes that is influencing every aspect of our life. And when it comes to especially the genomes, we know, and a good scientist is one who knows more and more about less and less. What he means about more and more about less and less is we have to understand the less and less of the microbes, and how they impact the crop physiology, their phenotypic expression, their fitness to the environment, productivity, and adaptability. I think it's a beautiful book, and congratulations, Dr. Nataraja Karaba. I would certainly like to have a copy of this book, probably without payment, in a lighter weight, and look forward to that wonderful contribution. And I would request Dr. Aparna, Dr. Nataraj Karaba for making your presence felt. I know it's a very odd time for Azhakanandam, but still he has been awake and for us ready to deliver a talk. I welcome Dr. Tiku, the Doyen of Horticulture, especially tomato breeding, and my good friend too, who have joined. So it's always a pleasure to be with all of you.
I was on mute. I heard you.
Thank you. Thank you very
Krishna,
much.
So
Congratulations.
before release, shall we request Nataraja sir to say a few words about the book?
Yeah, please.
Please, Nataraja sir.
Nataraj
Yeah.
Karaba.
Yes, sir. Can you hear me now?
Yes,
Yeah.
very well.
Yeah. Thank you very much, Dr. Krishna Kumarji. Right now, I am in BR Hills along with my students for a field trip. I didn't want to miss this opportunity. Thank you so much for the kind words. Special thanks to Dr. Aparna as well as Nagrani for putting all the stuff together. Which, in fact, this is a talk which I delivered at your place, online lecture. So you made sure that this particular concept is printed. I must thank ATPBR, all the team, including Dr. Krishna Kumar and Aparna, for putting them in a printable form. So just to mention that this kind of work is very essential in modern years. We have breeders put a lot of energy and effort to put all the genes together in a genotype, but then some of these genes are not getting activated. They need an elicitor. So now we know that in human system, this kind of hologenome concept is emerging. Microbial genome also plays a very, very important role in activating traits, as well as Dr. Krishna Kumar just now said, G into E and into M, microbial genome, and their interaction, combined interaction, determine the phenotype. From this context, I believe this book would be highly useful to the readers as well as policymakers and of course, industry also. You can probably have a matchmaking concept. Every genotype may need a specific combination of microbial genome, we call endophyte, so that host genome should be evaluated thoroughly and exploited for the benefit of farmers and industry. And I would like to thank again Dr. Aparna and Krishna Kumar and Nagarani for putting my ideas together and bringing this particular book. I'm sure it will be useful to multiple people. Thank you. Back to you, Dr. Krishna Kumar and Dr. Aparna.
Thank you.
Thank you, sir. Thank you, Rahul. Now I request Rahul to please play the video.
So,
So Rahul is playing the video.
yeah.
Thank you, Rahul.
Thank you
Now
very
I
much.
request Krishna sir to please take it over.
So I would not like to take. Thank you very much, Nataraj Karaba. What is the price of the book approximately? That this thing? Any idea?
Kitna, Rahul? So 500.
500. Okay. So ATPBR will this thing, and as well we will share it in the different WhatsApp group. I would also request the Azhakanandam to share it among the US friends so that it can have a wider access and also persons who can benefit, because these are all very unique books that you don't find every day persons contributing. And my congratulations to, on behalf of ATPBR, to Dr. Nataraja Karaba. And I would not like to take much time, but I welcome Dr. Kasi Azhakanandam to take over the floor. Is it okay, Dr. Aparna? We will request Dr.
Yes, sir.
Kasi
Please,
to
please,
this
sir.
thing and share your thoughts. I would request those of you who have joined also to keep your this thing mute, so phone mute, so that we don't have any kind of a disturbance. And please feel free to use the chat box to air your points of view, which will be compiled at the end of the day, end of the seminar or talk, and then point-wise answers could be provided. I would request also Nagrani to note the questions raised and the answers given, so that it will be in the fitness of things to see that when we summarize the talk, it would be nice follow up to say what were the questions that were raised and what was the answer that was shared, and it will be a wonderful event. Thank you very much, sir. The floor is yours, Dr. Kasi.
Thank you, Dr. Krishna Kumar. And first of all, I want to thank Dr. Nataraja for the wonderful book. I'll definitely buy a copy of the book, and also I can disseminate with other colleagues and so on here. I want to thank.
Can you also share it in the American Association of Plant Physiologists or some society? If you can just write and then put it to them. I think it will be very nice that those who are interested will see to it.
Yes, yes, I can. I am a member of the different society. I can share it with the secretary.
Yeah, thank you.
So again, I would like to start thanking Dr. Aparna for this opportunity to present today. And it's been great associating with your organization. It's very impressive. The work you have been doing is phenomenal, and most of them are voluntary and helping a lot of young students and scholars and breeders. And this is awesome. I think your association with all well-known people speaks a volume. And I hope that I could also contribute in the future. I'll start with my presentation. Let's see how it goes, and hopefully I'll cover go through them and then see how each one of you can connect and also do some work in the future. Already you're all doing it, but I thought it would be really good for us to know the challenges and also what are the solutions existing out there. So let me share the presentation. Can you see my presentation?
Yes, sir, it's very clear. Just click
Okay.
at the hide button at the bottom, so then this bar doesn't
Yeah,
disturb.
I'm just looking for the pointer. Yeah. Oh, this is anyway. It's okay. It's
Hide
not
at
a
the
full.
bottom. Hide at the bottom. You see.
Let's see.
That bar, can you hide and
Yeah.
yeah, that
It's
bar,
okay now.
two bar.
No. At the bottom, you see the stop sharing and hide. There's one pop-up.
Oh, yes.
Yes. Perfect. Yeah.
Okay.
Please go ahead.
Okay, great. So I think, as I mentioned, I think Dr. Krishna Kumar gave a really nice introduction. This is very relevant to global community, in particular global South. That's the reason I chose this topic. Challenges and the solution for food security, nutrition, and environmental sustainability in the global South. And I think most of the world really aware of all these challenges. There is a lot of information available, and there is a lot of solution being implemented. But I want to give an overview how it's really currently going on. Before I go there, you may be wondering what are all these pictures. So these are the crop I worked with the last 20, 30 years, starting with the rice and all the top, the food crop I was working on for several years to Mexico, and also I did work on rice for my PhD, and also several years at Tamil Nadu. And then in the US, predominantly worked on the corn and soybean for 17 years for insect control and herbicide tolerance and so on. So this is also a very appropriate time to thank Dr. Swaminathan for his outstanding contribution. This is a birth centenary year. And I know you all had a celebration last week in Delhi, and it is really appropriate to honor. I think any students here or research scholars, young people, should read more about him. As a scientist, as a visionary, as a humble person, how you really cared about, people of India and also global population eradicating poverty. And what I really like him, actually, I used to work under his foundation for a couple of years when I was doing my MPhil and PhD in India, part of PhD. He always says the sustainability. At the time, I did not understand fully what exactly he meant, but later on I understood. Yes, anything he does is care about not just for the farmers and people, but also environment. And I think this is very appropriate. And now everybody's talking about sustainabilities. This is the reason they're all visionaries. They know long time ago, people like him, people like Dr. Norman Borlaug. And I think there is a lot of coverage, and I was reading about this one. So I think if somebody is not really familiar with him, please read about it, especially for the students. So basically what I'm going to do is next 50 minutes or so, I divide into two parts. I would like to give an overview of the food security challenges, especially in Global South. What are the key challenges? And also want to go to the solutions. Obviously, we have been doing great with the breeding and so on. And on the other hand, the technological innovation really helping advancing the breeding. And also recently, integrating biotechnology approaches with the AI innovation. You cannot think some of things in the past, but now you can think and address the issues. You can really make a designed plan to really overcome the climate challenge and so on. And as Dr. Krishna Kumar mentioned, it's important to have integrated policies. Without that, we may not have the full benefit of all these technologies existing currently. And then we'll have a discussion at the end. And if somebody really need to ask a question, please stop me and you can ask a question. So basically, just to summarize a few key challenges for food security. I think we all know that population is the biggest concern, and currently we are 8.3 billion. In 2050, in 25 years, we'll be adding 1.5 more billion people, which is a lot of people. On the other hand, we need to increase the food production up to 70% based on the FAO. And 1/3 of the food production globally is wasted. This is really disturbing, right? Because including India, we waste a lot, and more than $10 billion they estimated being wasted each year. And this is a concern for globally. Maybe the issues are different in the global south versus north, but it is a big problem. Importantly, the cultivable land is shrinking due to the urbanization, and this is also, again, you can see it in India. And major crops like rice, yield is plateauing, and in fact, they need to increase like 30% in a couple of years. So ERI and other organization has been focusing on improving the yield. And moreover, agriculture is a fresh water. So 70% fresh water is being used for agriculture. And also, you know that even there are places we are struggling to have drinking water. So it is a real challenge. Besides that, we never thought 20, 30 years ago, climate change is the biggest challenge for us, but now it's real. And everywhere globally, agriculture is suffering. And it was predicted the time it reaches 1.6 centigrade above the post-industrialization, it will severely affect the crop productivity. But unfortunately, we're almost there. We can go into a little more detail because I wanted to tell you one thing. Yes, the challenge is big, but we are kind of constrained. We had to do sustainably produce more food from less. less land, less water, less resources. How can we really do it? That is the biggest challenge we have. Just want to bring your attention. We all know that population increasing, and that's a big challenge. To just say here, if you look at them, it is not going to decline anytime until 2080, 86, they say, which means we are going to keep adding more billions of people. In 2086, it's going to be 2050, it's going to be 10 billion people, which is lot of people. So we have to be really mindful. And also, surprisingly, most of the people populations are the increased population going to be in the global south. So which means we have more challenge. We have to really think through and then implement the good policies to address all these issues. Second thing I mentioned, land, because cultivable land is a really small portion. If you look at this one, most of the global surface is covered with the water, 70%, only 20-30% of the land is for all other things, including for agriculture, forest, and so on. If you really look at how much is being used for agriculture, it's very small portion. It's maybe overall like 30 million hectare compared to 141 million kilometers. In this one, if you really look at it, actually we spend only about 20% land for crop cultivation, and also 4% goes for the non-food crop. Basically, 16% of the land, which is 8 million kilometer square, is used for food production, which is very less, which means we have to really see how we can really improve the productivity within the limited land. In addition, climate change and other challenges, we have a war in different places, and then there's a lot of conflicts and so on. So there's a lot of pressure to really increase the food production. And another important thing is, and now I think slowly getting attention, the soil health is badly affected. Because of that, we have the reduced yield. It is especially prominent in the global south, because soil degradation contributes to almost 20 to 40% yield loss, which is a significant, and also is a predominantly, as I mentioned, sub-Saharan Africa is very high. And also you can see even in India, I think the salinization becoming a problem where the seashore is there, and we lose quite a bit of yield. So we already started with the less land. We are already losing the land. In addition, whatever the land we have, we are not having healthy soil, as Dr. Krishna Kumar mentioned. It is very, very important, right? We have to have a really good policies, and we have to make sure that we are using it, and also enriching the soil is very important. Why this degradation? Because during the Green Revolution, when we use high-yielding varieties, we dumped a lot of fertilizers, pesticide, which really now causing major issues. And so this is another concern for us to address. As I mentioned, the temperature or the climate change is a really, really big challenge. And as I mentioned, this estimated temperature 1.6 C is already reached end of last year 2024, and also early this year. And you can see from I think from post independence, this temperature is very increasing so much. It's the same thing everywhere, because of many countries' industrialization and a lot of climate affecting gas formation, greenhouse formation. So it is increasing, and it's going to continue for some more time until we do something concrete, like stopping the fossil fuel, for example, and also doing the sustainable agriculture can also reduce, and so on. So the challenges are enormous. Now, focusing on Global South, you can see Global South is the colored regions, Latin America and the Caribbean, Africa, Asia, Oceania. What is the key important thing is the population is already high. We have 7.2 billion out of 8.3 billion population lives in Global South. And also you can really see the food security is very, very severe here because of all the problem we just mentioned. So we have to really do something now to reverse all these challenges. And then if you really categorize them into three major buckets, nutrition is a key thing, because you can see that people in African country, even in India, we have a lot of undernutrition and also the micronutrient deficiency in young kids. And also, unfortunately, we have one side malnutrition, on the other hand, obesity also increasing. That's because of all this modern food and processed food and unhealthy eating habit, which cause non-communicable disease and so on. And then in terms of food security, we have a low agricultural productivity. Still, we are not compared to Western countries. That is because of various things, even the resource, the cost, and so on. And also, as I mentioned, the post-harvest is a severe problem. We lose globally $1 trillion per year. For example, in India itself, about $10 to $15 billion per year, which is a lot of money. And also, most of the global south depends on the food imports. For example, because of a tropical condition, we don't grow wheat, not in India, mostly for South African countries. They have to import the wheat like that. So there are still depending on importing food from other countries. And the environment is really in a dire need. We had to do something to address the climate change. As I just mentioned before, land degradation is another big problem. Water scarcity is another important problem. So you can see the challenge is enormous, right? So on the other hand, I think I'm hopeful by seeing the green revolution, when I was a student, and then seeing all the biotechnology advancement, and also seeing, advancement, and also for recent AI. I think I'm hopeful that we should be able to address all these issues. Hopefully, we can feed all the people, and without anyone going empty stomach because of all these issues. Just to show this picture, I was talking to the company CEO a few months ago. I was very disturbed to see that, I thought in Africa, every food is a scarcity, but they produce a lot of food. They also don't have the facility to store. They dump a lot of mangoes, fruits, everything in the roadside, which is sad because they just don't have the capacity to store, for example, for extend, some more month or weeks. Hopefully, this again, with the genome editing, with the new technologies, we should be able to address some of these issues. So these are the challenge. I think hopefully you've seen the bigger picture, and you can connect with where you are and see how we can do. And the solution, what are the solutions to address the food security, nutrition, environmental challenges? So obviously, yes, due to green revolution, I think we reversed the starvation, especially in India and other countries. And since then, the technological innovation is really making the big difference in all aspects. And recently, integrating the biotechnology approach with the AI innovation is very powerful. And I can see already institutions and universities and academic and various government organizations already looking into how we can really leverage all the technology to address these issues. And again, there are policies available for the food security, but I think it's time to have integrated policies so that we can see the end-to-end, how it affects one area to other areas, nutrition to food security to environment, so that we can really get the benefit of all these technologies. Finally, we'll have a discussion on the topic. So I just want to walk through. I'm sure many of the well-known people knows all the things, but if you're a student and maybe research scholar, just to give an idea, when I was small, when I was a student, maybe 80s, used to hear about a lot of talk about IRA rice, for example. Then only I came to know it's part of the green revolution. you all know that Dr. Norman Borlaug, who done a great job and worked closely with India, especially with Dr. Swaminathan and so on, played a major role to contribute in the green revolution. I think Norman Borlaug is from the US. I met him when I was a postdoc at NC State. Very humble man, very much care about the global community. For him, it's all about feeding the people. He said, if the person fed well, you can address half of the problem, which is true. And starting from the dwarf wheat, he's a maize breeder, but worked on wheat when he was in the STEMIT. And then this is the wheat came into Pakistan and also came into India. And this really revolutionized and addressed a lot of poverty during the 1960s, 70s, and so on. And for his contribution, they have awarded the Nobel Prize. He's the only first person in agriculture to receive the Nobel Prize. Very well deserved recognition. Then later on, 1980s, genetic engineering came. This is where I got really excited about all these technologies. And during that time, two things are very prominent. Marker arrested breeding is really helped a lot. And also introducing the traits, heterologous trait to the crop, especially for herbicide tolerance and insect control, is really revolutionized agriculture. And you can see three of the known people, Robert Frawley, Mary Del Chilton, Juan Montagu. These are the people played a key role. They all worked in different places, Europe to US, but they all able to present their studies in the same year, 1983, how gene can be transferred using agrobacterium. I think that revolutionized and increased the yield. You can see some information about that in coming slides. And then again, another new revolution came, genome editing. This partnership between Dr. Doudna and Charpentier played a key role in identifying the Cas9 protein, which can cut the protein from invading viruses. And they learned all the nature, how it does. They come up with the Cas9 genome editing tool, which revolutionized, even though there are so many other various genome editing tools available, but this one is simple, straightforward, and this has just changed the whole field, how we want to improve the crop and so on. And the key thing is this is just faster to market, less regulatory hurdles that made it even better. And then again, you all know that recently AI became a talk everywhere. I mean, of course, we have been doing the digital agriculture for a while, but with these two scientists, Professor Dr. Hoffield and Hinton, their foundational research and innovation paved the way for machine learning and artificial intelligence. I think they revolutionized thing. And also, if you use that, all the statistical tool really helped a lot to assist predictive breeding in agriculture. So it's all coming together in the right time with all this innovation and all the tools available. I think we should be able to address global food production issues successfully. I just want to go through the technological innovation quickly, because all the things changes, all things change a lot. That's because of we're able to sequence a lot of genomes. And now there is a Kew Garden in London. I think they have an effort to sequence all the flowering plots. They are very close to complete the work, which is really phenomenal. Not only that, every institute, even in India and China, they are sequencing every day some genome. It's really revolutionized because of that power and the tool available. We can create the climate resilient crop and also sustainable agriculture. Precisely, we can engineer the plant to really adapt a different climate and so on. I just want to quickly go through again. Agriculture has been there for thousands of years, but really the early breeding started 20th century onwards, and the field really picked up from 1950s onwards. and there's been a great contribution. On the other hand, the population growth is phenomenal, so not able to keep up. That's the reason we were looking for the new tools to really enable the plant breeding. For example, as I mentioned, the marker assisted breeding is very good. Early 70s played a key role. 90s genetic engineering, 2020s genome editing, and then 2020 is digital agriculture. I want to go through quickly what is a big difference. Basically, when you do the breeding, it's a purely crossing and looking at the phenotype, determining the performance of the traits, and then identify a good variety for cultivation, which takes a lot of time, which is not precise. But marker assisted, you can target, you can tag the trait you're interested in, you can move them and then trace them. It's a little bit more precise. And genetic engineering, you can just take any gene from any organism, we can enhance the crop of interest, which is great. By doing genetic engineering is good, but problem is a random insertion, so that you have to create a lot of transgenic event to identify a good one. But this is a challenge for us. But this genome editing just overcomes that one. Since availability of genome sequences, you can target any location in the genome. That's the beauty of this one. Because of that, you don't need to produce a large number of transgenic events to select a commercial transgenic plants. But combining all the thing with the digital agriculture, it is like magic, basically, right? So you can really do all the work, and then you can do a lot of work in the computer. Basically, you can do the pre-breeding, predictive breeding, and you can select the line which one you want to really go to the field to test them. So it's really enabling, and this is the kind of things happening. Marker assisted breeding, this is one of my favorite thing. I didn't work too much, but I worked a little bit. But the easiness and then the simplicity really helped advancing the breeding. And also, I think it's also simplified a lot of breeding and the field trial and so on. So I don't want to go through the detail, all the detail here, but starting from 1970s, starting with the RLF marker, but then now we are in the 2000 to 2010, this is where the genotype technologies really revolutionized. That's because the sequencing cost is so cheap, so that anybody can do the genome sequencing, and then you can really create the markers for whole genome and really help the breeding go faster and so on. And then 2010 onwards, the genomic selection and beyond is very, very important, because now you can do the whole genome, not only whole genome, now the pangenomic. It's basically you can sequence multiple crops of relevance and then compare how the trait behaves, and you can also study the structure of the trait and so on. So this revolutionized the breeding and is helping a lot. And again, I'm sure most of you know, this is just a process. Same thing how a breeder does. You take a susceptible resistant variety, cross them, and create the F1 populations, help them. You get the F2 population, then you can do the extraction and do the genotyping, basically do the PCR, and then identify the trait you're looking for, the phenotype, correlating your phenotype with the genotype. This simple process really enabled. And again, visually, if you want to do it, and here in the rice, again, if you want to cross or stack two different resistant gene, raise one for raise one and raise two, and you can, you have the one population is resistant and susceptible raise one, raise two, and then you have the second population susceptible raise one and resistant to raise two. So basically, you can create any number of population, and then you can do the same thing like PCR, and then look for which population has both your R1, R2 present. For example, here you have the resistant allele for this and the resistant allele for the other one. So this is simplified. Otherwise, it's blindly we have to do. It's a lot of work. That's the reason breeding takes a long time. But this one, you can do them in a very quickly, and then narrow down to the population you're interested in. So as I mentioned, I worked in Mexico for a short period, about 3 years. I was leading the crop transformation lab, but also involved with some of the marker as the breeding thing. Actually, we are creating the transgenic rice with XA21. As you all know that bacterial blight is a major issue for rice. And what we did, not only creating GM, we also stacked them using the markers to breeding, because we have the 6 different isolates. We wanted to identify the line which can be resistant to all the races. By combining, bringing the bacterial blight resistant genes, we are able to overcome most of the effects from most of the isolates. So this is again very simple. We train the person; he does all the work. So this is the magic of this, molecular astral breeding. And then when I was in Syngenta, our company also come up with a product called Artesian. Basically, this one is a very neat studies they've done, bringing the 13 different QTLs from different genotype to come up with a drought tolerance crop maize. And this one is really revolutionized. Because I used to see the product in the field, in farmer field. There was a drought, I think 2012 or 13, and wherever you grow the normal corn, you see the corn is not really, the grain is not fully filled like this. But whereas when we grow this artisan, this variety, you can see the fully matured crop. Basically, it increases 12% of yield, which is a big actually for corn. But if you have the rain comes, it doesn't affect. But if there's a drought, if there's a less water, this traits start kicking in, able to do this. And again, this is a molecular astral breeding, doesn't need to go through rigorous regulatory approval like a GM crop. So this is the advantage of doing the marker breeding and so on. And now, with all the invention, with all the sequence genome selection, basically you can predict, but may not correlate all the time in the field. That's a challenge with the computational biology. So now, with this paper, recently I just saw this paper, with all this improved algorithm, improve the statistical input, you can increase the accuracy of the prediction. That's what they're trying to do. If you're going to say 70% of the predictor line can perform well, but now you can say 95% you can get the high accuracy. It's all about accuracy, how much you can translate from your computation and prediction to the field. So there's a lot of advancement, which is really good. So by doing all the things, reduce the breeding cycle and the cost, and increase the precision in selecting the complex traits. And also you can stack the multiple traits, especially the disease and the drought. They are complex traits. It's not single gene counts, but it needs more than single gene. So the molecular marker really helps, and I think this is one way to really increase the yield and so on. And then genetic engineering, as I mentioned, this is where I spend most of the time for the last 20 plus years. And again, it's very simple. Really, we learn a lot from nature, right? And nature teaches a lot. Every technology you see here is mostly connected with the nature. And the simple agrobacterium, soil-grown agrobacterium can really transfer gene. Again, these 3 scientists are the one to identify, to transfer gene, you can use the agrobacterium, you disarm the native oncogenes and then insert the tDNA, which can be delivered into plant cell to get a normal plant. I used to work with Dr. Meridel Chilton. She's very innovative, smart, and sharp, and she used to work until 80 years in the lab. Now she retired. And it was great pleasure working with her, especially people like them. And fortunately, I know all these people. We meet them at the conference and so on. So it's nice to have somebody you look up and continue to do the good work. Because of they come up with this idea, it's revolutionized the GM crop, introducing gene into plant for various traits, various purpose, especially for the herbicide tolerance and insect control. The first crop was introduced in 1996. Since then, that time was only 1.7 million hectares, but now 2024, 2025, is closer to 200 million hectares globally grown. You can see that everywhere, it's been grown. For example, in India, the BT cotton, when I was there in Mexico, the BT cotton we used to import, but it's revolutionized. Unfortunately, later on, the other diseases attacked, the technology failed, but not because of BT technology. It's due to other one. So, especially in the Western world, when we grow all the industrialized agriculture, they don't have many people to do all the things. They use a mechanized. So they use these crops, and then the weeding and the application of the lot of pesticides all reduced. Actually, there's a lot of benefit introducing this crop to the global population. This work I did in Mexico. I was leading the crop transformation, the BT eggplant growing in Bangladesh, coming from Mexico. I was very happy that my team did a great job, and we identified the lead event and then sent it to Bangladesh, where they're growing, they're reaping the reward. By talking to the people work in the field, it's amazing product. I know this is amazing. We've seen in Mexico. But hearing from them is good. Actually, 2 months ago, I went to FAO conference Rome, presented the case study, how beneficial to the farmers in Bangladesh. It reduces application of the insecticide. It's increased the yield like 30 to 50%. And also now, because of this, people doesn't need to apply a lot of, they say they apply almost 70 to 80 times per season. That's a lot. And so imagine that how much chemical you dump on the ground. Not only that, imagine the person who sprays the human health and the environment, all the things. So there are advantages. So we just need to select the trait or technology for what is available. For example, I want to emphasize this is possible doing the genome genetic engineering, because there's no native resistance in brinjal. So you can't do much. So you have to bring in from outside. So using only GM, we can do. That's why I always say I would go with the nature. I go with the conventional breeding, do the molecular breeding. If it's not possible, we'll go with the GM. And that's the way we do. And I think that's been really successful. And also in terms of the addressing the malnutrition, I think you all know golden rice. Again, I'm very happy that Syngenta contributed the golden rice version 2, because even the version 1 is there, but the amount of the beta carotenoid is not very high. The vitamin A is not high. So we enhanced that one, and this increased enough quantity of the beta carotenoid. So it's really good. And thanks to these two professors, Dr. Ingo Portugues and Peter Bayer, who pioneered this technology and very passionate. He's another person who really wants to do something for the communities. He did a great job. Just imagine just taking the gene from different source, like from plant daffodil and also from soil-grown bacteria. We can insert in rice so that you can address vitamin A deficiency in the young children's. Which is good. As fortunately, it's been approved in Philippines, but still not yet commercially grown. Hopefully, it will be released and people can get the benefit. Another example from the GM is increasing the important carotenoids in tomato. Dr. Kathy Martin from John Innes Center did a great work, and she worked for many years, but finally now it's available in the market. Basically, this is very, very good, right? So people like to use a tomato everywhere globally. And then in addition to get the food nutrition, you also get all the essential, amino acids, essential, nutrients. And this is the kind of advantage of using the GM technology. It's not in the native form, but you can bring them from the various sources so that you can enhance them. Like that, there are so many examples. Just to summarize, what is the really beneficial? Yes, we grow a lot of GM crop. Definitely, it's increase the productivity and the biodiversity. Also, safety environment, and also for the people, and also it's reduce the carbon dioxide emissions and elevated poverty for millions of farmers. and then people associated with the farming community. So this is all good thing, but definitely it also has some drawbacks. And since I worked on, I know in and out, and we have challenges. Definitely, we as a scientist, we believe it's a good technology, but on the other hand, it's not accepted by the public easily because of the perception. Oh, you're modifying the plant, coming from different source, all kind of thing. And also, it's expensive technology. It takes minimum 10 to 15 years to commercialize. In the US, to commercialize single crop, we spend about $200 million. That's a lot of money, and it takes a lot of time. That's because of, not because doing the research, also regulatory takes like 4 to 5 years to get approval, so what. It's a longer timeline. And also another thing I'm concerned about is if you grow them for longer period, like in BT cotton in India, eventually insect pests get resistant. Or if it's a herbicide tolerant crop in the US, we also have the weed, super weed problem. So it's nothing wrong with the technology. It's all about how you use it. The same thing with the conversion breeding. If you use like IR8, is so good. Later on, it's becoming susceptible for disease and insect and so on. So if you are going to, that's another problem, the monoculture, they say. If you are going to use the same crop for continuously year after year for a longer time, eventually you plant, lose resistance, so on. So if they want to implement the technology, they can use it, but when the time comes, they need to take it out and then use another version or different gene or different technology and so on. But to overcome all these things, the genomic medicine came in the right timing, really. It's exciting for us working on this area, because what are the challenge we had, with the plant breeding and the GM technology, this is really doing the work. As I mentioned, genomic is not new. It's been there. People started working with the meganucleases, zinc finger nucleases, talent. But they're all good. They are demonstrated. Company worked on it. They have a successful thing, but very complicated. To make the construct is complicated. To do all the work is complicated, not efficient. Whereas the CRISPR-Cas9 technology came in 2012, really revolutionized. Just briefly, in nature, you have a lot of organism. We have bacteria. The bacteria also constantly attacked by viruses. In order to protect from the viruses, the bacteria come up with a mechanism. This protein, Cas9 protein, goes and chops the viral sequences, which means it cannot produce a functional protein. That means it cannot affect it. By doing that one, it every time chops, it leaves the traces in the genome. So it's kind of memory. It's like a vaccination. And then when a new virus comes, keep attacking, chopping them, and then able to survive. By observing the nature mechanism, how it's able to protect, that's how these two scientists, they are able to come up with re-engineer, reprogram this Cas9 protein, and also make it very easy using the guide RNA. All you need is, you need a Cas9 protein, you need a guide RNA to guide the Cas9 to genome where you want to specifically cut or make changes. So simple. Nowadays, even school kids can do. You just train them. The kit is available. You can buy it. You can do it. So that much easy. They made it very useful, simple. So what can it do? That's the key. Obviously, we have a double-stranded DNA in the plant sequence, and basically any changes you want to do, you have to either cut, yes, or you have to inactivate the base pair from the gene of interest. So you can, as it shows, yes, you can cut them and make any changes you want. And this is like a Swiss knife. Basically, it has a multiple function. You can just change one base pair. If you don't want to go and do a lot of things, you can just make a modification. And then you can also do the replacement. Yes, you want to change the amino acid, so you change the base pair. You can make small changes, big changes, no problem. Or you can also insert the gene. Like instead of doing the GM, you can put it in a Cas9 cassette construct and then insert the gene of you are interested in. Why it is important, as I mentioned, you can insert in the plant genome where there is no effect on the plant. Imagine the GM has an issue when you insert them, then it's a random, it may affect a native housekeeping gene, which will hinder the plant performance. But here we don't have. This is where all this genome sequence information and all the computational power comes into play. We can choose the location you want to put it in, and then we can do safely and without affecting any native genes and so on. So basically, it's a very versatile tool to make a modification to the plant to whatever you want. You want to enhance the quality or abiotic stress or biotic stress, or the drought tolerant. Yeah, any of the things you wanted to do. So again, this is a versatile tool. It's revolutionized. I've been in research for 30 plus years, but even to keep up the literature is very hard. That much revolution, everybody. The globally, everybody's doing it. Now China's leading this area in agriculture. And I'm very glad that the Indian government also identified 40 crops. Actually, I was talking to Dr. Bansal recently. He was telling about all the exciting work going on in India. I think this is a way to go, and it's a good technology. Again, within a short period of time, there is a wide range of work is going on, different purpose and application. This shows that it covers pretty much all the thing, agronomic values, improve the yield, and then also food and feed quality, biotic stress, herbicide tolerance, abiotic, and so on. So basically, within a short period of time, it's increasing, and there's diverse. But that's a fantastic advantage of this technology. You can pretty much target anything you want to do. And this is just to show that the crop is a wide range of crop, like cereal. They're all genome edited. Some of them are commercialized. Some of them are in the process of commercializing. You have all the major cereals are here, potato and vegetable crop and fruits and so on. So it's a revolutionary technology for plant biology, plant breeding. And also the first genome-edited tomatoes commercialized in Japan is called GABA enriched tomato. Again, increase the nutrients. And I think more crops are going to come. In fact, the US, there's a company called Pairway, just a few miles away from where I live. They work on many different crops, including the vegetables and fruits and so on. And they also commercial a few years ago. Basically, they did the editing with mustard greens, which is a pungent. It's not very palatable, but people like this one, so they edited. And then it's very niche market, not a big one, but this shows the power of tools. So you can do, and then they also had a seedless blackberry. Now they're working with other blueberry, many berries they're working on. So basically, it's a very powerful technology. You can use it for your advantage. And then in terms of the crop, I mean, in Africa, I think they are doing the sorghum, gene-edited sorghum for this disease-resistant, the witch weed problem in sorghum. And then companies also, they are advancing. Bayer or Monsanto, they have the shorter corn. The corn grows so tall, which means it takes up a lot of resource and so on and timeline. But here now, they shorten the corn, but the yield is the same, nothing changed, which is pretty good. I think they are in the commercial now. And also Dow, not Dow, Corteva, they have the Maxicon. So there are a lot of things going on with this technology. These are all advancement, but again, overall, the plant breeding or coming with the product is a timeline. In the industry, what we always care about, who can go to market first, how fast you can deliver the product to the farmers or grower. So yes, you can do the genome editing, you can do the GM, you can do the normal marker breeding. Still, the timeline is long. But now in Syngenta, we combined the double haploid corn breeding, double haploid technology with the genome editing. Basically the goal is you create the normal, traditionally you insert the gene, and then you do many selfing, backcrossing. It takes many generations. But if you are using the double haploid corn technology, if you have the line where you can edit. Basically, when you edit that one, you can quickly double the chromosome using this constant treatment. Basically, you can cut down the number of cycles, and then you can directly go to the homozygous generation in the second generation itself. Basically, you can eliminate all of them. So all is about selecting this Merle line and make the edit and then cross with your, whatever the events, whatever the line we want to do it, back crossing. And here is we can avoid all things because we want to have the homozygous for trait. We want to do it very quickly. That's what they did. So now this can cut down the timeline significantly so that your product can go to market much faster. So also, as I mentioned, another beauty is about genome editing is people have globally accepted most of the countries. And in fact, you see that the green one, including India, they also consider this as a kind of breeding technology. And it's not like going through the GM regulatory process, which is good because you can get the editor line, you can much faster, you can commercialize, which is good. So this is the simplicity. The technology is already efficient, and then it can use it. You can really bring it to the farmers much closer and quickly. These are the advantages with genome editing. Okay, now I'm in the final one. Artificial intelligence. I think, do I have enough time?
Yes.
Okay.
Yes,
great.
please, sir.
Okay, so everybody's aware of this artificial intelligence, so I don't need to say much, but just wanted to clear. Yes, I think digital agriculture, you can say it's kind of umbrella. And then using all the digital technology, drones or GIS system or sensor, all the thing. Underneath, you have the